Can you hear me? Yes, can you see us?
No. I just see a black screen.
Okay, hold on one second.
Turn camera. Okay, it should be us, right?
Okay. Hold on one second here. How come I've got nothing?
We're having technological
it worked before.
Here. Okay, so what is that? What is that? No, not sure.
You can hear me. How come I can't see this? This is the camera.
Normally it would put us like
down here, or whoever's talking goes on the big part. Sorry guys. Bear with me.
Just
no, that's not going to be it. I used this last week.
What if you restart? Is that turn camera?
Oh. How embarrassing. It's good. Okay. There we are. There we are. Can you see us now?
Yes.
Okay.
Hi, Kelly. Hi, Dory.
It helps to turn the power button.
At UMA, and if it's too noisy, I can shut my microphone off and just type questions. If that makes it easier.
Let's see how it goes.
Seems okay right now.
What we're gonna try and do, and I don't know if this is gonna work, is we're gonna try and show some PowerPoints. Charlie's gonna talk for a while. I don't know if the camera is gonna pick it up. Okay, so
yeah, we'll see. Yeah, we'll see if we can see some slides here.
See if you guys can see this. Can you see this?
I'm just seeing you.
You're just seeing me.
Okay.
Yeah. That is unfortunate.
Yeah, no big deal.
Sorry. I'm not sure how to do that. Hold on a second.
Enable content. So there's a way to like share a screen or
yeah, I don't know.
Slideshow.
Well, that's just gonna bring it into like a bigger, the whole thing.
Okay.
Oh wait, is that online?
Okay.
That's on a web browser.
Oh, okay. Yeah.
Should I agree to that? I know
Sure.
it's your stuff.
Oh yeah, that's fine. Yeah. Right. There's nothing in there that's proprietary.
That's really fast.
That's slow, isn't it?
Okay. I don't have any
camera.
Yeah, okay. One more thing here.
Monitor view. Insert. Is there a?
I want the camera thing. Let's see if there's something in the camera. Just give us another minute, guys.
Webcam controls. What settings?
No screen capture. Yeah, there's no
setting.
There's no. Sorry, I should have asked you about this earlier.
No, that's okay.
I would have thought.
Culbertson photos. Sharing clips and
Maybe.
well, yeah, maybe.
No. You have to like make a movie and then.
Oh, okay.
All right. No screen capture. Search screen capture. One more thing. Live stream video is an image.
Which one appears on your computer? So it's live cam. Huh? Is that?
That's that thing.
Okay.
When you start with the live stream video, you can use your live cam for a video. Yeah, it's just a test.
Okay.
All right. Okay. We're not going to have pictures, guys. Sorry. It's just going to be us. Okay. So, Kelly and Dory are here. That's great. Welcome to our Google Hangout with Charlie Culbertson. Charlie's a microbial ecologist and hydrogeologist with the U.S. Geological Survey in Augusta. Before coming to Maine, Mr. Culbertson spent many years studying water systems and the bacteria that affect them in California. His work has made him an expert on selenium and arsenic cycling in groundwater and surface waters. In addition to studying arsenic, Mr. Culbertson has worked on many local projects establishing monitoring for nutrient pollution and other markers for water conditions in the state of Maine. Hi, Charlie.
Hi guys.
Can you start maybe by giving us some background about yourself and how you became involved with the USGS?
Yeah, absolutely. So I back in the good old days, I graduated from got my bachelor's degree at San Francisco State in San Francisco back in the mid '70s, and at that time, like many recent grads, I had no clue what I wanted to do. I had a general degree in microbiology, and actually had joined the Peace Corps and was going to head to Africa to do fisheries biology program with the Peace Corps, and about that same time my father ended up getting ill, so I decided not to leave the country, and anyway, I heard about an internship program at U.S. Geological Survey out in Menlo Park, California, and I applied for that and ended up getting it, and so the project that I was going to work on was to do work in San Francisco Bay, looking at. Did we already there? Okay.
No, it's just the noise.
Okay.
Yeah, we're still there.
So the project was going to be to work in San Francisco Bay, looking at the nitrogen cycle. And some of the slides I was going to show you is kind of what the various parts of the nitrogen cycle are composed of, and Pete tells me this is an environmental science class. So, but at any rate, so I said, "Oh, sounds like a pretty good project." And so when you go about studying something like a nitrogen cycle, there are various tools that you have at your disposal, and so I started working in San Francisco Bay, looking at a process called denitrification, which is an important process. Still, I mean, has huge climate change impacts today, and the process of denitrification sort of takes nutrient pollutants, if you want, that have primary nitrate, and so it's has a big agricultural component to it. So denitrification is a process whereby nitrate is converted ultimately to nitrogen gas, and along that pathway, and that's done by an enzymatic reaction by bacteria, and along that pathway, certain things are emitted, and one of those things is nitrous oxide, N2O, and which turns out to be a pretty big greenhouse gas in today's current global change climate, and so one of the big issues continues today, that N2O in the atmosphere stays is a pretty big degrader of the ozone layer. So so my project was to measure rates of denitrification in San Francisco Bay, and the way I chose to go about that was to add, and we and Pete can attest, we in microbiology use different tools, different inhibitors and things like that to sort of give you an idea of how these processes work. And one of the tools that you have for for studying microbial processes is these different inhibitors, and the inhibitor I chose to use was acetylene gas. Acetylene, as many of you probably know, is a welding gas, and it's it's been around for a long time, and it's actually used to be part of the early atmosphere, and it has volcanic origins, and so it's been around for a long time, and so Precambrian or early bacteria have been exposed to this gas early on. So it's in terms of the genome for bacteria, it's been around for a long time, and their ability to deal with this gas is very well documented. So and what acetylene does is block. So as you have bacteria that are feeding away on nitrate and converting it along the pathway to nitrogen, giving off nitrogen gas, acetylene stops that process at the N2O level. So if you can imagine, and one of the screenshots I'm going to show you is if you put mud in a flask and you add nitrate and you add acetylene gas to it, and you cap it and you shake the heck out of it, and you look at that over like a week. You do like what is called a time course experiment. So you're measuring from time zero. So you know how much acetylene you add, you know how much nitrate you add, and you know how much at the beginning, how much nitrous oxide, N2O, is there, and then you go along daily and measure these things, and so you're looking at the increase of, because acetylene blocks the process at N2O can't be degraded, so it sort of builds up in these flasks. So by the rate at which that builds up gives you an idea of how fast. So you sort of extrapolate back to the real world, and it gives you an idea of the rate of denitrification. Bacteria also convert nitrogen through a process called nitrogen fixation, also a very important process in ecology and environmental sciences, is bacteria being able to fix nitrogen from the atmosphere and convert it to ammonia or nitrate. So the process goes both ways, and it's and sometimes the same enzymes that sort of facilitate this, and other times it's different enzymes. So and acetylene blocks that. So in the process of doing my experiments, I was probably 6 months into my work. I noticed that in a certain set of flasks, that all of a sudden the acetylene was disappearing. That these are sealed flasks. Acetylene is a hydrocarbon, it's a gas. Up to that point, nobody thought that anything could happen to it. And so I went and told my supervisor about hey, the acetylene is gone in these flasks, and he's going, well, you must have had a leak. There's no way that it can disappear. So I went back out and did the mud, grabbed my mud again, did the same thing, and 4 days later, boom, acetylene's gone. And bought new stuff. I changed everything that I could think of to prove to him that there was not a leak in the system. But the other interesting observation that I made at that point was that in the set of flasks, and these are my controls, that in that set of flasks, the mud turned jet black. And one of the pictures I'm going to show you was I mean, you sort of have a series of flasks that have normal mud in them, sort of a grayish brown mud, and then this one set of flasks, jet black. And you open it up, it stunk to high heaven. So we're all of a sudden we're thinking about, wow, this is something new. Nobody's ever seen this happen before. This acetylene gas. It's been used for decades before I came along, and but the point of this story is that as often as that occurs in science, you go in with a plan, and you go in with a project in mind and a problem in mind, and just by happenstance things can change. And in this case for me, my project went from looking at denitrification and nitrogen cycle in San Francisco Bay to looking at hydrocarbon degradation, microbial hydrocarbon degradation in the natural environment, and that's what I ended up doing my master's degree on, was looking at bacteria. I ended up isolating these bacteria that grow exclusively on acetylene gas, and they make all kinds of interesting compounds, so the point is that you sort of get directed, and I mean science sort of dictates just by if you happen to be lucky enough to observe this, or it happens. So you just never know, and so what happened with that course of those sets of experiments sort of set me on a different path. Although now I've come around full circle and I do nitrogen cycle stuff in Maine and other places, but that started me looking at anaerobic hydrocarbon growth, and at that time it was we had a lot of the Voyager spacecrafts were going off, and people were looking at extra sort of the idea that life could exist on other planets, and in writing my master's thesis, you start asking the questions. Well, why acetylene? Why do they have this ability to grow on this gas? And why do they have enzymes that enable them to do this? So you start looking at reasons for why an organism would is it defense mechanism? Does it get carbon out of it? Does it get energy out of it? There's got to be a reason why microbes do things in the natural environment, and at any rate, so in writing my thesis up, I started coming across some journal articles from astrophysicists talking about, well, life could exist on these other planets if such and such exists. We have acetylene, we have methane, we have all these other compounds out there, and they go, but there's no documentation that things can live on things like acetylene. I'm going, here's a little master's student going, well, wait a minute, I have a bacteria here that does this, so we wrote a couple articles for Origins of Life journals and others, and it sort of got to start getting it picked up by atmospheric people, and in fact one of the people that reviewed one of the early manuscripts was Carl Sagan, and because he was one of the early people that talked about life on other planets, and actually one of the moons of Saturn, Titan, is one of the areas where they hypothesize that if there's an extraterrestrial environment that would work, Titan would be it, and because it mimics a lot of early conditions on Earth. But anyway, so that got me going for quite a long time, and then sort of my career sort of went from there, from working in San Francisco Bay mudflats to going to hypersaline environments, working in closed basin lakes, desert lakes, high desert lakes where there's input through groundwater or freshwater inputs into the lake, but there's no flushing, there's no exit of the lake. So the only exit from lakes is is through evaporation, and it turns out, I mean, these environments are called extreme environments because they tend to be very high pH. They tend to be very high saline environments. You know, and they tend to have all sorts of pretty amazing metals and other chemicals in them that nothing else lives in them except bacteria. So it's a great place for microbiologists to go look at processes. And at that time, I was within the U.S. Geological Survey. I was in a program called the National Research Program, which is a pretty cool program still going on within USGS. It does basic research on, you know, as long as it's under the guise of water. You can you're free to look at whatever you want to look at, and so we chose to look at these extreme environments because they perhaps would be analogs for early Earth and analogs for other you know, life on other planets if you want to go that far. But in fact they are. These extreme environments are. You look there, and these bacteria are very, very, very old. So these are the kind of places you want to go to if you're interested in bizarre organisms. This is where you go.
The edge of life.
The edge of life, exactly. And so or actually two of the big issues in the western volcanic areas are arsenic and selenium. And I got myself exposed to selenium early on in the 70s in California, and actually it's being repeated today. You know, water wars in California, and there, you know, as you know or you may know, that California is going through you know, a prolonged drought. You know, almost 8 years now of drought-like conditions, and so. You know, I just remember, as you know, being in San Francisco, seeing like pictures on the news of, you know, people in Los Angeles filling their swimming pools and watering their lawns, and here we are in San Francisco. You can't take a shower for a week, and you can't flush your toilets, and, you know, severe water conservation. So one of the great ideas that the Army Corps of Engineers had was that oh well, we can transport fresh water from Central California and bring it to San Francisco, and that project was called the San Luis Drain, and it got about, I don't know, 5 years into it, where they built, you know, massive canals to transport Central California water and Sierra snowmelt water up to San Francisco, and the project for some reason ended up being defunded by Congress, and so the San Luis Drain made it as far as just on the sort of just, you know, near Monterey, but out toward the Central Valley, made it about that far, and about 80 miles short of San Francisco, and it terminated in just by, you know, I don't know if it's by plan or not, but it terminated in this wildlife refuge called Culbertson Wildlife Refuge, which turned out to be on the major Pacific flyway for migratory birds. And about 2 years later, I mean, so it says you have all this ag water, all this water being sort of transported up to you know, San Francisco, and just stopping right there. And it turns out that people started noticing two-headed ducks and just all sorts of bizarre things going on to
Two-headed ducks.
Absolutely. But so all sorts of waterfowl issues were being observed with these birds in the Culbertson Wildlife Refuge, and so we got involved and went down to start looking at what could be the issue down there, and it turns out it was selenium, which is a metal, you know, it's a naturally occurring metal or mineral in rocks, and it was being sort of weathered, dissolved by the water, you know, as it was being transported north. So one of the things I do, and Pete introduced me as a hydrogeologist. I do a lot of what's called geochemistry, and really specifically what I do is biogeochemistry, which so the study of geochemistry is the study of rock water interactions, looking at whether it's groundwater or whether it's surface water. It's a study of water mineralizing or weathering rocks, and so whatever's in the rocks can get in solution, whether it's in groundwater or if it's surface water. And in the case of California, it was just the serpentine rock at the surface that was being weathered, and selenium was coming out of solution and generating, getting in high concentrations in these waterfowl or eating vegetation that is loaded with selenium, and then the rest happens. So that's geochemistry. Biogeochemistry is the, you add biology to it basically. So and it's really you add bacteria to the geochemical processes. So you're looking at naturally occurring bacteria that affect the mobilization or the mineralization of these metals, and so it turns out bacteria are very important in selenium mobilization as well as arsenic mobilization, and so I got involved in looking at microbial processes that affect selenium to begin with, and then we from there sort of evolved into looking at arsenic, microbial biogeochemistry of arsenic in various environments in the West Coast, and about that time I moved to Maine. I mean, I spent 20 years in the USGS out there, and I had recently gotten married, and we both didn't. I mean, my wife had a job at University of Connecticut, and I was in California, and neither wanted to move to each other's locations, so we came to Maine. And as luck would have it, or not have it, Maine has a huge groundwater arsenic problem, and so I immediately was able to dovetail myself into ongoing arsenic work in Maine, and in Maine the issue is groundwater arsenic. So I mean 75% of the population in Maine gets their drinking water from drilled wells now, and probably a conservative estimate of that, 20% of those wells have arsenic in them, and some levels, very high levels of arsenic, that rivals any sort of world concentration of arsenic. So arsenic is a very big issue in Maine, and so I began looking at sort of the temporal and spatial variability of arsenic across the state, and you start coming up with a general picture in terms of, well, geology is pretty important here, and so why are there clusters of high arsenic, like let's say in around the Penobscot region, or over in near Acadia, or down in Buxton Hollis. I mean, so there are these various regions of high arsenic clusters, and it turns out that there's arsenic-bearing rock in these areas, and as we drill into these arsenic-bearing rocks, then you're going to just by, again, geochemistry, geochemical processes and microbial processes, you get arsenic that gets mobilized into your well water, and so that's one of the things I'm now doing in Maine is looking at those processes and trying to come up with different alternatives to how we get around this issue, and so I'm looking at household water treatment systems, how they function in terms of removing arsenic from your drinking water, and what are the constraints on those systems, and why do they work sometimes, why do they not work other times, and so that's something I've been involved in the last 10 or 14 years since I've been in Maine. I was at a meeting earlier this week, and just curiously, one of the questions when I'm working with a homeowner and giving them their arsenic results, and sometimes it's not a very good
Not what they want to hear.
not what they want to hear, and so I get a question a lot about, well, gee, my grandparents and my parents' grandparents and beyond have been drinking this water for years, and there's never been a problem, and I mean arsenic is I mean, sort of from the health perspective, can create a lot of different health issues, from bladder cancers to cardiovascular issues to you name it. It's kind of implicated in a lot of things. But so the question they always pose me is, well, why is it a problem now? And the answer is that it wasn't until really the early 1950s or maybe 1960s where we went from, for most people that had wells on their property, had dug wells on their property, to technology now that allows you to drill very deep holes into bedrock. And so the switch there is that before, these dug wells were not getting exposed to where the arsenic is, and now we are. And so that this workshop I went to was to maybe talk about going back to using dug wells, so I mean, one of the problems with dug wells is that they're shallow, and they tend to run dry sometimes. So you have to be cognizant of where you put these things. But now there's some pretty cool new engineering. I mean, a typical dug well in the old days would really just be like a 4 or 6 foot hole that was down about anywhere from 10 to 30 feet, and you sort of, you luck of the draw in terms of how much water you got. And so now there's some pretty cool engineering designs that enable you to capture water from a pretty broad area and stay above the arsenic and other metal issues. So this kind of, in terms of environmental science, for some of you folks, I mean, these are areas that are pretty fun and wide open, I think, to look at. So that's the drinking water part of my job I've been doing. As Pete mentioned, I've been doing some other stuff, and part of my other research is working up at Acadia National Park, and this is where my nitrogen training came back full circle now. And one of the big issues up there is the estuaries are becoming, people are just noticing that they're seeing more algal blooms and sort of these not very picturesque things that occur at a normally very beautiful area like Acadia, and they're wondering why all of a sudden are we seeing these things, and it all has to do with urbanization, and in that area it's all about more and more people are building. You have more septic tanks coming online. You have you're just raising the ante in terms of more nutrient transport or more contaminant transport to natural systems, whether they be lake systems or little streams. Ultimately, things end up in the coastal waters where we live, and that's just water flows downhill, and that's where it's going to go. So we were asked to start looking into first of all, how bad is the problem and what can be done about the problem. And so that's one of the ongoing projects we have up there, is to look at again, rates of denitrification. I come full circle to doing what I started 35 years ago, sort of studying these processes in Acadia, and trying to give park managers a tool to go to city planners and say, hey,
this is how much the system
this
can take.
This is how much the system can take. If you plan on putting a new housing development in this area, we need to have certain. There's got to be some way to mitigate the potential issues of nutrient transport. And so it's as an environmental scientist, it's pretty fun to be involved in collecting that data and transmitting that information to people that know how to make a difference, and people that politicians and park managers and city planners and all those people are involved. But it's the science that drives that whole thing.
Or should drive.
Or should drive the whole thing. Good
people
point.
are involved.
Good point. And so it comes down to politics. Pete's exactly right. And it's just like another issue we have in Maine is atmospheric mercury deposition. In virtually every freshwater body in Maine has a mercury advisory on it. And why is that? It's not that we we have a lot of mercury in our environment, natural environment here locally. No, our mercury issue comes from Nebraska, comes from Iowa, comes from Illinois, through coal, through coal, and through burning dirty coal, and we happen in Maine to be on the what's called the exhaust pipe of the U.S. And it just turned out just the normal flow of the jet stream brings most of that contaminated air right over the top of us, and empties, sort of goes out to sea right at Acadia, and so you get atmospheric deposition of, not just mercury, but nitrate through burning of fossil fuels and burning of gasoline. Sulfate, sulfur dioxides coming also from burning of fossil fuels and gasoline. So all this stuff has been deposited on us, and all the lakes and streams, most of them in Maine are acidic also, and it's because of this atmospheric deposition. We know what the solution is. Shut it off at its source. Put scrubbers on these exhaust pipes that are burning coal. The technology is there, but the will to do it at this point, and it all comes again is politics.
Yeah. Well, the interesting thing too is those systems in the Midwest. The reason they built the smokestack so tall was because they needed to meet regulations that were local. So in order to make the air clean where they were, they put them up tall, and then it came over to us. And it dissipated, but it still ended up falling down, 700 miles to the east.
They knew exactly what they were doing.
It's all engineered to do that. Yeah.
But it's
the law, what is the law? What are people, willing to negotiate as a solution?
Right.
It's a huge part of environmental science.
Yeah. It totally is. It's a great example of cause and effect, and so you still have these very powerful coal lobbies in Western Virginia, you name it, that are very putting these strippers on the exhaust is not cheap, but it's also doable, and so it just comes down to the will and the political drive, if you will, the political climate to do that. So you as or we as environmental scientists, those are the challenges that we face. One other thing I'll talk about that is kind of fun to me, and I just came from a WebEx a couple hours ago talking about this. And it's the topic of harmful algal blooms, a harmful algal or an algal bloom. Probably everybody has seen them occur, and if you're around the water very much, you see, typically in the summertime you'll see, algae sort of, the water will turn green, and it's very unsightly and whatnot. But so, as it turns out, the frequency of these algal blooms over the last 2 decades, 3 decades, have been on the rise. Not just in the U.S., but globally on the rise. And what what's the one thing that is in the news daily these days is global climate change and global warming so is there a connection between global warming and the frequency of these algal blooms? And it's hard to refute that there's not. When you look at again, as an environmental scientist and a microbiologist, which is what I am, when you look at things that control how these blooms form, it's first they need a nutrient source, and that's going to be either phosphorus or nitrogen, and most of that comes from agricultural input sources. The other thing they need, all algae need, is sunlight and heat. And so you put those two things together, and you look at areas like California that's going through long-term drought, or people in the Southwest. You look at areas like China that have high ag inputs, and so global climate change doesn't just single out one area of the globe. It's global, and so we're seeing these things globally, and they're on the increase. But the reason this is even more of a concern to me is that these algal blooms produce toxins. They produce and you either know somebody or probably read somewhere where people will have their dog swimming in a lake that has an algal bloom, and all of a sudden the dog dies an hour later after getting out of this lake, and it's from swimming in these lakes that have these blooms occurring in them, and so these things they're called cyanobacteria, blue-green algae, which is a more traditional way to describe them, but they're not algae at all. They're actually bacteria. They're actually more aligned with bacteria, even though they have chlorophyll. They belong to the bacterial classification, in that they look genetically more like bacteria, except they have this pigment and
they photosynthesize.
And which is why they are so adept at they can sit dormant for decades. They can be in a desert environment that goes through either monsoonal seasons, or they can sit dry for decades, and you add a little bit of water, and these things, boom, they spring to life, because they have these little specialized cells that enable them to survive being desiccated for who knows how long. But as soon as you give them the right conditions, these things spring to life, and they produce a variety of neurotoxins, and in fact, the neurotoxin, it's a suite of neurotoxins they produce. But in a class, when you look at, one of the ways of assessing how bad a toxin is as an environmental scientist is to assess what's called an LD50 assessment, where you take a bunch of mice and you expose them to this toxin, and it's the amount of toxin that kills 50% of those mice. That LD50 is a gauge of how toxic something is, and the toxins that a lot of terrorists are sort of interested in these days is ricin. It's produced by a mole. I think ricin produced by a mold spore or something.
I thought it was a castor bean.
No, it is. Okay,
That's a plant one.
Okay,
that's
yeah.
a plant one. But there's plenty of them out there.
Plenty of them out there. And then
that are microbial.
And then cyanide is another one. But the neurotoxins that cyanobacteria produce, or orders of magnitude much higher.
You guys are probably most familiar with this when they shut down the shellfish.
Exactly.
The shellfish beds every summer. Those are from algal blooms in the ocean.
Yep.
Right. The so-called red tide.
The red tide is.
And that's also increasing in frequency.
Yep.
And
absolutely.
You eat these shellfish, and I think the last case I read about was some lobsterman found a barrel in the ocean, and there were all these mussels growing in it, and he took it home to his family and served those mussels that night, and the whole family ended up in the hospital, and nobody died. But
luckily,
it's amazing how strong these toxins are.
It is
because the mussels concentrate the algae by filter feeding.
Yep.
So
yep. And it's most of these toxins aren't degraded by heat. I mean, a lot of people would think, well, if I cook them, it'll be fine. Well, no. In some cases, you're actually making them worse.
Yeah. The other example you're probably familiar with is, I don't know if you guys ever been to China Lake. But China Lake is a very shallow lake, and it's filling in. And for years, they've had trouble with the water quality there, mostly just from people fertilizing their lawns.
Yep.
So it took them years to get people. Well, you probably know more about it than I do.
Well,
that's a local classic.
And that's a really cool one, because as Pete said, so it's been happening for years now. It's been shallowing, and so the planners have known, that okay, well, we have these inputs into China Lake. So what they've tried to do is cut off these little streams and sewer inputs into China Lake. And they've been pretty successful at doing that. But yet the blooms continue to occur. And so as an environmental scientist, you're going, well, why? So they're cutting off the source of contamination to the lake, but yet these blooms in some cases are not only continuing, but they're getting worse. And so it's a really cool environmental problem. and so what we think is going on is that there is this internal. So let me step back one step. That the again, algae need two other than sunlight and carbon, which they fix out of the atmosphere, the photosynthesis. They need two things. They need nitrogen and phosphorus. And in typical freshwater systems, phosphorus is always a limiting nutrient. There's usually, because these things, again, they have these specialized cells. If they have ammonia around, if they have nitrate around, they can feed on that. But if there's none of that around, they're so well adapted, they can pull atmospheric nitrogen out and grow just fine. So in freshwater systems, it's the phosphorus that controls everything. So in China Lake and in other lakes in the Northeast, we're starting to figure out is that there's this internal phosphorus cycling that goes on, and it's driven by what's called redox, which you can kind of trace everything back to, again, it's a microbial problem or internal microbiology problem.
For us, anyway.
For us, yes. So what happens in the summertime? The water heats up, the lake gets like thermally stratified, and so meaning that there's sort of two lakes in one. You have the surface water biota that's there. That's plenty of oxygen and plenty of fish and all the good things you want in the lake. But in the bottom water, it tends to go what's called anoxic. So microbes in that area consume. They're doing what they do, and they consume oxygen, or actually the oxygen disappears by them doing what they normally do. It goes into oxygen depletion in the bottom water. But what happens is as oxygen starts to decrease in this bottom water, it's called redox. As the redox drops, phosphorus gets mobilized. So as oxygen drops down to a certain concentration, just by what's called reductive dissolution, and it happens a lot in the real environment. With arsenic is another example. But as that happens, phosphorus gets released and gets mixed back up, and boom, you have these blooms, and then. But nothing really, so they incorporate the phosphorus into their cell material, but then at some point the bloom crashes. Where does that go back? Where does it happen? It gets settled back down in sediments, and then waiting for the next process to happen. So you have this constant internal cycling for phosphorus, and that's what is in fact driving a lot of these blooms. Even though we've cut off the ultimate source of the contaminant, it still is continuing to have an effect. And you can make that,
because phosphorus doesn't go into the atmosphere.
there's
It's
no-
contained
yeah.
inside the aqueous and
Yep.
solid
There's
system.
no gas release. Like for nitrogen there is, but for phosphorus there's not. And
Yeah.
so yeah, I mean, you can make this as an environmental person, you can make this leap to looking at greenhouse gases, for example. As we release more CO2 or methane in the atmosphere, just because we cut off the source now, I mean, that stuff is up there for a long time, and it takes and N2O that I started talking about early on is one of the most long-lived of those. But CO2, nitrous oxide, and methane, once it's released from the terrestrial or aquatic environment here, it's up there for a long time, and it's
It cycles through.
it cycles through. Yeah. So cool. So anyway, that's kind of what I am involved with at USGS and what a lot of environmental scientists in academia and elsewhere do, and it's a pretty exciting field.
Cool.
So I encourage you guys to. Sure. Yes.
Okay, my first question was when you talked about the well water.
Yes.
And the artesian wells compared to the dug wells. Is there anything that engineers can do for the people that do have the artesian wells now to help, like some kind of filter that they could put on the pump itself that pulls that water out of the ground?
Oh yeah, absolutely. So
For arsenic specifically?
Yeah, because you were comparing how they're doing, engineers are working more on the dug wells now
Yes.
to try to go back that way and
Yep.
get away from the artesian wells, which they thought, 25 years ago they thought those were better because of you don't run out of water
Yeah.
as
Right.
much. So now you have this huge population that has artesian wells, and they don't have the dug wells anymore. So instead of having to go back that
Right.
way, is there something that those engineers are working on to help those people?
Yes, absolutely, and this is another good area of research as an environmental scientist. So first of all, an artesian well, I may have misspoke. So an artesian well is a well that actually it is fed by spring water. Is more of a shallow type well that kind of has a pressure head and it flows by itself. So I was trying to make the distinction between, and that's generally is a very, if you have an artesian well, those are pretty. You want to hold on to those babies.
Yeah.
So we went, 50 years ago we went into drilled wells, into drilled bedrock wells,
Where you have to pump.
where you have to pump, and that's what you're talking about. And so yeah, there are, and as it turns out, I mean, so arsenic isn't the only problem we have here, and the other problem you may know of people, or you may yourself have radon issues, for example, in your water, and that's usually a granite. It's usually the source rock is granite, typically for radon, and then uranium is another big concern that we have in Maine, in certain areas. But the good news is that all of those, and specifically arsenic, depending on the form that arsenic, I mean, in the real environment, in the natural environment, arsenic exists in two forms. Sort of an oxidized form it has lots of charge on it, lots of electrical charge on it, which makes it very easy to remove. It's sticky, and so there are a lot of materials that you can design, and that it's on. I mean, the field right now is going crazy trying to come up with new materials that work very, very well for removing not just arsenic but other contaminants. The other form that arsenic occurs in the environment is the reduced form, and it's usually done by bacteria that sort of in the process of consuming the oxidized form, they make the reduced form. So that form also occurs pretty commonly in the environment. That has no charge on it. In the natural, from pH 4 to pH 8 or 9 in the most normal natural waters, It has no charge on it, so
Very soluble.
it's very soluble and much more difficult to remove. So the challenge for environmental engineers is to come up with ways to address that issue. And one of the ways that currently people do that is by installing a system at the house that oxidizes the heck out of the water. So they're either through crudely through bleach. Bleach is a very high oxidant. That's why it works so well in detergents and laundry detergents and whatnot. So it oxidizes things and makes them very, very polar and very easy to remove. So, but to get back to your question, so it depends on really the concentration of arsenic in your well water, and there are certainly places in Maine. Like eastern Danforth, Maine, areas around Blue Hill, where the concentrations are so high that normal technology right now is fighting to come up with a method. I mean, these systems, like a homeowner may install this on their house, and they're not cheap, by the way, either, and it may work great for 2 months. But then the system gets just saturated and you can't regenerate anymore. So you have, so you're constantly trying to stay ahead of the game in terms of what type of treatment systems you put on. And so this is
Arsenic's a bit tricky because unlike iron or manganese, it's a little easier to reduce it. So it's a little easier to put it in the bad form, right?
Yeah.
And it's harder to keep it in the easily reduced, easily expelled form. It's trickier than the other metals, but you can find systems. And around Augusta, there's a not as high a concentration, but there is arsenic in the well water around Augusta. And Charlie's been working with the public health officials and the toxicologists at the state to test well water around Augusta and to get people who have concentrations above the advisable limit to install these systems in their home. And if you have a well and you look, and you can find these maps online, right? And if you are on well water, it's really a good idea at a minimum to take a look at your area and to see if there's arsenic in the water in your area, and if so, get it tested.
There's a great.
It's just a one thing you can do.
There's a great website at Maine CDC. If you go to maine.gov, or I think if you just Google Maine CDC, and it's just gone up in the last few weeks. They have these risk assessment maps for arsenic and radon, fluoride. They're doing it for a number of things. So by the state, it just gives you. It's a really cool interactive. Like database, and you can kind of see areas that you're at risk, that you might be at risk from. And Pete mentioned iron. I mean, iron is not in itself a toxic mineral unless it gets way up there. But iron is one of those things that again, as an environmental scientist, it reacts with everything. It absorbs stuff and
clogs up your well.
You makes your laundry brown or red. So it's one of those issues that, again, especially in filtration systems, that's a big player, and it's one of the reasons we're considering other alternatives for homeowners for drilled wells, because if you're unlucky enough to have high iron and high arsenic, then you're just doubly doomed, and it's just really hard to, and then the problem is exacerbated by people with young kids, and all these questions you have as a homeowner to make these decisions about, well, should I drink bottled water, should I bathe my kids in this water? All these things you need to be concerned with. And again, as an environmental scientist, that's where you come in. So it's So it's pretty. I mean, there are a lot of different avenues. Public health avenues you can go into. There's basic research. There's all kinds of really cool. For me, I mean, if I were coming into this field today, it's much more clear to me now than it was 35 years ago in terms of, gee, what do I, where do I want to go? So anyway, I hope that answers your question.
Yes, it does. I just have one more.
Oh, sure.
To rush back to a conference. You were talking about the bacteria that kills the dogs when they're swimming in like lakes and ponds.
Yes.
And could that hurt humans?
Oh, yeah, absolutely.
How do you know if it's there? How would you know
can
that
see
it's
it.
in the lake? Is it something that's just silent and you never know?
No,
until
you
the dog passes
know very. So it's I mean a pretty. I mean, if you want to like see some pictures of these things, if you just Google harmful algal blooms. And in fact, Lake Erie this summer was a good example of in that case they shut down the drinking water intakes from around one of the areas of Lake Erie, but because of these blooms. But you can look at satellite images and just see. I mean, this is a pea soup green or blue green color, and it's very when the blooms are occurring and they can come up really quickly. No, they absolutely have a huge human impact, and in fact, I'm working with a group at Dartmouth, neurology people, and now they think there's a link between these cyanobacteria blooms, these blue-green algae blooms, and Lou Gehrig's disease, ALS, which we've heard a lot about this summer. But turns out only 5% of ALS is genetic, and the rest is environmental.
Huh.
So it's these kind of things.
That's more of a chronic exposure.
Yes, exactly.
Because I always think of them like if you can see the algae and it's really thick, then because all toxicity is concentration based. So it's about where this stuff is at a level that'll hurt you. So if you can see if it looks like pea soup, then you probably don't want to drink it or swim in it.
But one of the other challenges for environmental scientists is trying to understand things like that, because we still don't know. We've studied these things for decades. We don't know what makes them go from producing a toxin to not producing a toxin. Same organism, same bug.
Right.
So
that it may be some nutrient eliminating thing where they're going to try and kill whatever's alive so they can get more nutrients out of it. Who knows? Or maybe it's completely not related.
We don't know. We don't know. So it's so there's a lot of pretty cool areas to look at. That's a great question. It's not just limited to animals. It's really the human toxicity. That's what gets that's what gets the politicians to take notice.
So I have a question. Can you stay for a few more minutes, or do you have to go?
The conference is going to start back
Okay.
here in 9 minutes. So
Okay.
I have to rush back in.
Okay.
Go ahead and ask
No,
the question.
go ahead if you've got another question.
You got a quick question, or?
No, I didn't have any questions.
Oh, okay.
Those were the only two.
Oh, okay. Well, Chad Hill was wondering about groundwater and aquifer resources being sold out of state to commercial interests. And he wanted to know basically, is this a resource that we need to conserve in Maine, for groundwater rights, the Nestle and Portland Spring, or or is there a risk about these resources in general being depleted by selling it in bottles all over the country
and world? That's a great question. To take my government hat off and my citizen hat on, I have a problem with that.
Yeah,
definitely. Especially in the changing climate we have. The climate predictions for the Northeast is warmer and wetter, which means we get a lot of our storage, a lot of our groundwater storage, and replenishing these aquifers comes from snowmelt.
right.
And
The rain runs off.
Rain runs off.
Slow percolation.
I think it's a huge issue and a huge question. It's should we be getting rid of this I think the unknown of climate change, because we know the people's wells go dry in the summer. We're starting to see the higher frequency of bedrock wells going dry. So
Interesting.
so that's something that he's right on with that question.
Short term versus long term.
Short term versus long term. In California, again, it seemed.
Oh, they're at the poster child.
Yeah, this is
Yeah.
And I mean you look at historically, globally, I mean more wars have been fought on over water than maybe even religion. I mean, it's just amazing. So
Can't do without it.
No,
Can't do without it. Yeah. Interesting. He has another question, and I know you have to leave, but he read a paper regarding thermal imaging to detect anomalies in a specific river, the Menestakaig in Holton, and he was wondering if you knew anything about that, what they found, or do you know anyone who's involved with that?
That was me.
That was you. Oh, you know somebody who's involved with that because you did that one.
I published.
Were there anomalies?
Yeah. It's
what was that all about?
So one of the challenges you have we're looking at. I mean, so when you're looking at nutrient transport in a system, either it's very obvious where it's coming from, for like little small tributaries and sewage drains and the like. But another big input into our rivers in Maine is groundwater, shallow groundwater, and that's shallow groundwater is a conduit for whatever happens to be in the water. I mean, be in the groundwater at the time. So what we were interested in doing was taking this infrared or thermal imaging stuff that had been tested at very little scales, and what infrared does is to just look at the difference of heat. So you just look at heat anomalies, and so by doing these aerial surveys, you can look at very large sections of a river that were otherwise you'd have to go either by boat or by walking with a temperature sensor going behind you and looking for these small changes and temperature changes that might be an indication of where groundwater is coming into the system.
Okay.
So
you know the temperature of the groundwater.
So either you do this, so you take advantage of the temperature of the stream water and temperature of the groundwater. So either you do this in wintertime, where groundwater is warmer than your receiving water, or you do it in summertime where the groundwater is much colder. So, but nonetheless, the technique works the same. You're just looking at changes in temperature.
Yep.
So it's very so. This technology looks at as little as a hundredth of five hundredths of a degree
from aerial.
Yeah.
Wow, that's impressive.
So we were able to put a GPS on a camera and fly. I got many hours of air sickness doing this, and we just flew these rivers, and you're really just looking for these little hot spots.
Huh.
And then once you identify them,
so you did it in the winter.
We did it in late spring.
Okay.
Which kind of
ice out.
Yeah. Or early fall when leaves are off. So you just want to sort of maximize your interference issues.
Get to the ground.
But we did for the medocs. We found a number of thermal anomalies that. Either there were spring inputs and then you sort of go ground truth those once you've identified it from the air.
Okay.
And
then you can test that water to see what's coming in.
You test that water. It's a way of focusing your science. So now then you go to that area and put your piezometers or your ways to measure groundwater into an area where that there's an input of either deep groundwater or shallow groundwater into the system. And,
I never heard of that before.
We did that project with the Holton Band of Maliseets, looking at really they have a big issue with nitrate and
so looking for agricultural runoff that's not in obvious streams and things like that.
And they had these recurring summer blooms, and they're going well. And we already did a previous study. We eliminated the sewage treatment plant as one of the culprits and determined it was ag. It was some of these ag inputs.
Non-point source point.
That's a really important term. Is these non-point source inputs to an environment. So that's a good drive home message there. So, well, thank you.
Thanks for hanging out.
Thank you.
Absolutely, sure.
Take care. let me just
Yeah,
stop
we
it.
just published that.